The plasma membrane, the cytoplasm, the nucleus with its DNA, the endoplasmic reticulum, the Golgi apparatus, the mitochondria, and the microtubules. The cell, in short, the basic unit of life. It's been on Earth for more than 3.5 thousand million years. In all that time, it has evolved into an extremely complex machine that even today we do not understand. Then, in essence, what is life? What makes different a dead, bland and boring object like, for example, a bunch of planks from a living restless organism? I mean, for what we know, only living organisms have a metabolism, replicate their DNA, divide and grow. Then, wouldn't it be fantastic to create life from scratch? In the Christoph Danelon lab, we are doing exactly that, building an artificial cell with a metabolism that allows it to replicate, divide and grow in the simplest possible way. By putting together inner molecules like lipids, proteins and DNA, we hope to be able to build our own personal Frankenstein, what in synthetic biology is called a minimal cell. But is this actually possible, or just pure and simple science fiction? Well, we have some ideas to make it true. In order to equip our minimal cell with a metabolism, we use the so-called pure system, a perfectly defined mixture of enzymes that transcribe and translate DNA into proteins. By encapsulating this machinery inside vesicles, we would be plugging in the engine of the minimal cell and exerting a genetic control of all processes inside the cell. Now, living organisms also replicate their genetic information. Our strategy harnesses the power of viruses in DNA replication. By using their machinery, we expect to be able to control when and where the DNA would be duplicated. But wait a minute. Can you imagine a cell duplicating its DNA again and again in an endless cycle? Definitely, the cell has to divide to produce its offspring and proliferate. We approach this by creating a protein ring that constricts and pinches off the vesicle, hoping to fuse the membrane and create two single independent cells. But again, we cannot make to divide the vesicle indefinitely if we do not make it grow. For that, we introduce the necessary genetic information inside the vesicle to synthesize the phospholipids that spontaneously would get integrated in the membrane. And voila! We have a minimal cell. Do you still think it's science fiction? Pass by and ask, or even better, join the lab and make the minimal cell possible.